Hello, the guest today is Hao Xi, a PhD, class of 2020. He commercialized his PhD research into a startup. It's a Series A startup now. He's going to share with us how he built his startup. So Hao Xi, what does your startup do?
Hi, nice to meet you, Tony. So I work at Canvas, and we are a microbiome therapeutics company. So we essentially design and build a complex microbial consortia as a therapy to treat various indications. And currently, we are focusing on cancer immunotherapy to try to improve the efficacy rate of patients receiving immunotherapy.
Well, what is the microbiome?
Yeah, so everybody has a microbiome. It's essentially the collection of microorganisms that lives in your gut. And there has been a lot of studies over the past several decades about the gut microbiome. And we, as a field, are now realizing that the gut microbiome is very important for a lot of the physiology of humans. So your health, your digestion, your mood all depends on what your microbes are doing in your gut.
Gotcha. So let's say I eat a banana and the banana goes into my tummy. Well, what does the gut microbiome do with that banana?
So the microbes will probably chew on some of the more complex fibers that doesn't get digested in the upper digestive tract, and they will produce various metabolites that then gets absorbed by the host cells, your gut cells, that then does various things for your body.
Oh, interesting. So how does the gut microbiome, the digestive system for the banana, how does that affect the immune system?
Yeah, so that's a very interesting link between the gut microbiome and immunity. So there are actually lots of immune cells in the gut. So they are essentially surveilling what's going through the digestive tract. And so people have realized that actually these immune cells are really in tune with the microbiome. So they kind of know what's going on in the gut. And in turn, the gut microbiome exerts effects on the immune system. And so that kind of changes how you respond when there is a pathogen in your body. And similarly, when you have cancer, your immune system oftentimes also needs to be involved. And so that's where the gut microbiome comes into play. And what people figured out in the past couple of years really is that not everybody who receives immune therapy when they get cancer responds to it. What's more interesting is that people have done clinical trials and they have essentially looked for these people who responded to therapy, and they took their stool. The stool is essentially a collection of the gut microbiome. And they took these responders, they took their stool and gave it to non-responders, so people who failed immune therapy. And what they found is that you can actually convert some of these non-responders into responders, meaning that if you modulate the gut microbiome, you can help some people to respond to immune therapy better.
Oh, wow. Let's say there's a person who has cancer. So what's the process that patient would be cured of cancer?
Yeah, so usually you will probably first you go to see a doctor, and they will probably get you on various types of therapy. Back in the days, it's lots of chemotherapy, radiation, but more recently, people have developed immune therapy, meaning that you would essentially give the patient a drug to tell the patient's immune system to go loose on the cancer cells. But the problem is not everybody's immune system responds to this drug. And so sometimes, even when you take the drug, your immune system doesn't know to go for the cancer. And so what we are learning is that if you tune the microbiome in the right way, you can actually restore this response so that when the patient takes the drug, their immune system will know that, oh yeah, I should go after the cancer now.
Interesting, huh? So this isn't just for stomach cancer then, even though that's where the gut microbiome is?
Yeah, so this is actually has been shown in many different types of solid organ cancer. And the idea is that the immune cells from the gut, they're not stationary. They actually travel to other sites in the body. And so this effect is not local, it's actually systemic.
Gotcha. So then immune therapy would be more effective as a result.
That's right.
What is immune therapy like?
So the common ones are pembrolizumab. That's a very well-known therapy. And so the patient would usually get it through IV infusion. And so
Okay.
it's kind of you go to the hospital and you get it as an infusion. And yeah, and then the drug does its wonders for a fraction of the people.
Gotcha. Huh, that's interesting. And then because the gut microbiome fits better now, then
Yeah,
it'll work for even more people as a
that's right.
result.
Yeah.
What's the process of building up this collection of stool samples?
Yeah, so that's also a very interesting question. So we actually work with MD Anderson Cancer Center at University of Texas, and what they have done is that they have screened a bunch of donors. So these are people who responded to immune therapy. They screened their stool and they tried a bunch of different person's stool, and they actually identified this person that we call super donor. And so this is a person whose stool converted or helped multiple other non-responders to respond to this immune therapy. And so at Canvas, what we're doing is trying to replicate this magic as a reproducible, manufacturable drug rather than relying on this person to donate all the time. Everybody's gut microbiome is very dynamic. And so you could have a Chinese food and it might change a little bit. You might go travel for a week and it might change a little bit. It's very dynamic. And so the idea is that what you want as a therapy is something that's stable and reproducible that you can make at a large scale. Yeah.
Huh, interesting. So like the statistics work out, but is it sort of like a black box where we don't really understand what's happening within that stool sample, which causes it to happen?
Yeah, I think there's still a lot of open questions, but I think as a field, we have a very good understanding of who is there. And then what we bring to the table at Canvas is that we have this imaging technology that allows us to ask not just who is there, but also where they are relative to each other and relative to the host cells. And so we're using that to really get a better picture in a very literal sense of what is happening at the gut microbiome interface.
Like how precise is it? Like the donor A, the best fit for patient C, but the donor B might be an even better fit for patient D. Like how precision medicine is it?
At this point, since as the field is very, very young, we are not quite there yet. And so what we are doing is identifying this super donor and using it for multiple different patients. And I think as we go forward, as we get more understanding, we can imagine a scenario where you have multiple different types of drug products for different various cancer indications. One combination of microbes might be more effective for a particular type of cancer, but there's a lot of work to be done there.
Gotcha. So this super donor that the MD Anderson has found so far, it's just for one specific type of cancer, or is it for many types of cancer?
They have actually tried it on a bunch of different cancers, and it does seem to work across different types.
So this super donor is very, very good then.
Yeah, it is. It's quite super.
Have they have they tried tracking what happens to the patient after they take the microbiome? Is there like an increase in T cells and B cells? Or does the T cell is a little smarter as a result? Like what happens after they take it?
Yeah, there are increases in particular immune cell populations, but again, these studies are quite macroscopic, I would say, at the moment. So it's hard to dig into the specific mechanisms. I think to get there, we still need a lot more data, many more clinical studies.
Gotcha. Okay, that makes sense. So we know it works, but we don't really know exactly why it works then.
Right.
Gotcha. Okay, that makes sense. That's quite exciting time to be in this industry then.
Yeah, it is. The microbiome has seen some up and downs, but I think it is a very interesting field and there's a lot of problems to be solved. And so we're glad that we're tackling it.
Yeah. Wait, so I'm trying to picture what happens inside the patient's stomach after they take the pill. So they take the pill. Does their gut microbiome grow a couple extra things in a certain way?
Yeah, so certain strains from the drug product will fill niches that are not filled before in the gut, and over time they'll start to do their magic. And so you can think of the super donor microbiome pill as a complete ecosystem that has a particular set of functions when they work together. And so when the patient takes them, these bugs will engraft in the patient's gut and they'll start to exert their therapeutic effects.
Gotcha. So I'm like picturing like an ecosystem, like a natural ecosystem forest, and it introduce like an invasive species and it changes the ecosystem as a result. Is that the dynamic?
Yeah, you can think of it that way. It will certainly change the recipient's microbiome and everybody will respond a little differently because this biology is usually very messy. But the end result is that your immune system should start to respond better to these immunotherapies.
So is this like a permanent change, like after the cancer treatment?
I think some strains will probably stay for a while, but the microbiome is quite dynamic, so it will probably change over time from everything else that happens in your daily life. So
just eat some more Chinese food, and then it will change as well.
That's right.
Huh, that is fascinating. It's like a vibrant ecosystem in there. Is there like a different like dimensions, like you add a couple more birds to the ecosystem, or you add a couple of bugs to the ecosystem, or a couple more plants?
Yeah, you can imagine more different crosstalks between different people, if you will, or different groups of microbes in this case. These gut microbes, they don't work alone, so they're aware that they're in the community. So a bug A might produce something that bug B needs, and so on and so forth. And so they really works as a community that communicates with each other and exert effects collectively. It's a balance.
Oh wow! Is it like 10,000 different species living in there, or is it like 100 different species?
Each individual person usually have a few hundred species, and oftentimes the abundances are such that there are many abundant species and then there are many very, very low abundance species as well.
Gotcha. And then like in the natural ecosystem, in the nature, you have like a hierarchy of like this is predator, the
Mhm.
lions at the top, and
Yeah.
then underneath you have like a hierarchy of prey, and that smaller prey eats even little prey. Like, is there a hierarchy in this ecosystem?
There probably is, but to really get to that level of detail still requires a lot of technical work, including using our imaging platform, because it's very challenging to just try to understand the system by just looking at essentially the parts list, which is what most people can do so far. So what we're trying to do here at Canvas is trying to see how they are assembled together relative to each other. So spatially looking at them to try to get a
Oh,
better sense of like how these communities are put together.
so there's like some upstream ones, and then there's
Yeah,
some downstream ones as
right.
well. So location matters too.
Yes, yes, yes.
Oh wow! And this is in the small intestine, which swiggles around, or is this in the big intestine, where it's just like a big
So
chunk?
it's all over. There are the microbial load is generally lower in the small intestines, and then it starts to increase, and it gets higher and higher, and the highest point it reaches is in the colon.
Gotcha.
But there are lots of immune cells in small intestine, so even though the microbial load is low, doesn't necessarily mean that the small intestine is not important. And so when we study the biology of gut microbiome and host interaction in mouse models, for example, we look at all parts of the digestive tract.
Gotcha. Oh, that is fascinating. So,
Right.
but the stool comes out at the very end, though, right?
Right.
So,
Yeah.
stool is the end,
Yeah. When it comes out, yeah, it's at the end.
but it'll collect stuff upwards as well.
Yes, yes. It will collect stuff and it will evolve as it goes down because the environment of the host is also changing as you go from small intestine to cecum to the large intestine.
Okay.
Yeah.
So as the banana goes through the whole body, it collects some at the small intestine, then collects some more in the big intestine, and then collects some more, and then it pops out.
Right. At the end, it's probably not banana anymore, but yeah, that's the idea.
Okay. Gotcha. Huh, interesting. Oh, this is very cool. Wait, so you said that so it's not just a pill that you're making, you're also making a machine too?
Are you talking about a microscope?
Yeah, the microscope.
What we have been doing at Canvas is so far using commercially available microscopes to generate these imaging data. But the challenge with commercially available scopes is that they are usually point scanning to get the spectral aspect of the information that we need. And the downside of point scanning is that it's very slow. You're building an image point by point, quite literally. This point scanning strategy is very slow, and we like to get lots of data at Canvas. And so what we decided to do was to build our own instrument so that we can collect data in a much faster rate. And what we ended up building is a light sheet microscope that allows you to collect spectral information with a light sheet so that you get, instead of point by point, we're getting a whole plane instantaneously.
Interesting. How much microscoping are you doing? Like, isn't there if there's like a super donor from MD Anderson, that's just one guy, and then you have one stool sample, and is that all you have to image? Or like what? How much are you imaging?
We're doing a lot. So it's not just that one person's stool. We have done a lot of mouse models, for example, and so all of these experiments contain lots of information that we want to get them out of from the sample.
Gotcha.
And so there's never a shortage of things to image.
Interesting, huh? So back to the overall like large scale picture then.
Yeah.
Are you like what phase of trial are you in clinical trial wise?
Yeah, that's a great question. So we are actually getting ready for our first clinical trial in October this year, and so
Oh
that's
wow.
very exciting. Yeah, that one will be for one of our programs in addressing ICI-induced colitis. So this is one of the side effects that people get when they get on immune therapy. Many patients develop colitis. So this is inflammation in your gut, and then some of them will eventually have to stop the immune therapy. And what we found is that, again, similar idea, if you modulate the microbiome, you can tune the immune system such that you don't get colitis as often. And in fact, in some of our collaborators' trials, the amount of people that got colitis in their trial was exactly 0, which is quite astounding.
Oh, actually, this is great then. So yours isn't just making immunotherapy more effective. You're also making immunotherapy have less side effects too.
Exactly, yes.
So of all the different immunotherapies and all the different cancers you could do, how did you pick the colitis one to target first?
So I should say this is the first clinical trial we're doing because colitis is one of the biggest side effects that happens when you get ICI. But then the second trial we're doing is for the first indication that I talked to you about, which is to try to improve the efficacy of immune therapy.
Gotcha.
That one will start later in 2026.
This is great. So you have like a whole pipeline of trials. It's not just one trial.
Right.
So,
Yeah.
they always talk about platform in biotech. Always platform. We're not just single drug. We're in platform. Well, what's your platform?
Yeah, so our platform is really a imaging-based platform, and so it helps us identify many different species of bacteria in a single experiment. So that's kind of the genesis of Canvas. And what we found that it's very useful for discovery, so to understand how the microbes interact with the host. But what we also learned as we got ready to become a clinical stage company is that imaging is also super helpful when you look at manufacturing. And so we actually can use the imaging technology that we developed to assess the products that we're making to make sure that all the cells are happy, that they are doing what they should be doing when they get packaged into a pill.
It's great. So the microbiome, it's like a very good use case for this technology because there's so many different species in it.
Right.
And like a pill, like before it gets packaged, that's also a really good use case because there's a lot of complexity there too.
Exactly. And that's actually a very important point, but perhaps sometimes not as well discussed, which is that the field has been lacking technologies like this to really assess what exactly is going into the pill. So you might still have the microbe in there, but they might not be as effective as if you could actually measure it and tune it and then make it better.
So back in the old days, there's like the simple molecule drug that's like easier to measure, but now it's biologic, so it's more complicated. So you gotta have a better microscope now.
That's right. There's also more complexity. We have many species in a single pill, so you have to really have the capability to measure all these different species in a single go.
Gotcha. And these are the things that the patient's swallowing down. It's going to transform the gut microbiome. So you got to make sure it matches.
Yeah.
Gotcha. Well, it'll be the same pill for every patient that's on the drug then?
Yes.
It's not uniquely tailored to like my gut. I ate a lot of Chinese food the last couple of days, so my microbiome might be different from the next guy. But this is like a very specific. It's the same one for everybody.
Same pill, yes.
Okay, gotcha. So that's why. But it's important to make sure that the pill has everything you need in it.
Right.
Oh, this is great. Wait, so this imaging tech is very important then. So it's a plane. So it can do a bunch.
Yeah, it's like taking lots of snapshots for entire planes in a single instance. So you take one plane in one exposure frame, and then you take the next plane, rather than trying to build a plane point by point.
That makes sense. And this is important because it's not uniformly spread apart. The gut microbiome might have like some species here and some other species there, so you have to do a whole plane.
It's also important, particularly for the microbiome research field, because as you can imagine, individual microbes are very, very small. But tissue is actually quite large, even for mouse, for example. And so we really need a technology that has the requisite spatial spectral resolution as well as the throughput to image large pieces of tissue.
Gotcha. Wait, so it's 10 to the negative 6 meters? Is that the range you're working on? Micrometers?
A typical E. coli cell is like half a micron by 2 microns.
Yeah, these are pretty complicated molecules. So a couple micrometers is like good enough resolution then. And the goal is to just identify them. It's like let's name one of them, like species A.
Yeah.
Is it like the same species A as anybody else's species A, or is it like just completely unique to the individual?
That's a great question. There are certain shared strains between people, but there are also many unique strains to each individual. And I use the word strain very specifically because strain is a very fine taxonomic resolution. So if you talk about species, we probably share many common species, but species is a big umbrella when you talk about bacteria. So there's many different within a species.
That makes sense. So micrometer level is important then because then you can tell which strain you have as well.
Yeah, we need to have the resolution to be able to actually tell cells apart.
Okay, gotcha. Okay. So it's not just like looks like an elephant, it looks like a giraffe, and we're good enough. But you have to tell what type of elephant it is, what strain of elephant. Gotcha. So this sounds more like a physics problem than a biology problem, Hao-Si.
It is certainly a physics problem. I think nowadays science is really cross-disciplinary, and so we really try to use all the tools that we have at our disposal to solve the problems in front of us.
So what was your process of going from your PhD to this company? What was your PhD on?
Yeah, so my PhD was very physics heavy. So it was mostly developing this imaging technology that allows us to do these highly multiplex measurement of the microbiome. And so I was wrapping up my PhD at the end of 2020, and that was peak pandemic, if you recall. And so that gave me a lot to think about in terms of what I wanted to do next. So I was exploring many different options. So there are kind of two main factors that led me to decide to launch Canvas with my co-founders. One was that my advisor, PhD advisor, came from a very entrepreneurial lab during his postdoc. he has a lot of experience or exposure to that side of the world. And so he was telling me all about it. And he was suggesting that I really consider spinning out Canvas. And so that was factor one. The other factor was when I met my co-founder, Matt Chang, and he's a doctor by training, infectious disease doctor. And we talked a lot about the technology, what it could do. And he was quite convinced that there's a unique opportunity here. And in my mind, my advisor, PhD advisor, will always think that what we did was really important. So, as a good advisor would. So when Matt explained to me his rationale, it really kind of clicked something in my mind that, wow, there's someone outside of me and my advisor that thought this was interesting that we should pursue. And so with that, we decided to launch Canvas and give it a go.
What was interesting about it? Did you like see a big indication that has a big blockbuster sizing, and then you saw it as like the step by step, or were you like, this area is sort of is like broadly interesting, and we should explore something within that area? Like, how specific was your interest?
I think at the time I saw the potential the technology could do, because the technology I built was really to allow us to visualize the microbiome in a much better way. And the microbiome is not just in human gut, it's everywhere. It's in the ocean, it's in the soil, it helps plant growth, it helps agriculture. And so I really like the idea that here's a technology that has many, many potential applications. And so why don't we try to build it up further and see what it could do?
Gotcha. Oh, that's really interesting. So you saw the technology had a lot of future applications, lots of things too. And it specializes in gut microbiome because it's like a very interesting area with a lot of long-term impact.
Yeah, so that's also a great question. I think the gut microbiome has a unique connection to human health, obviously. And so there's a lot of interest in trying to use what we know today to help other people. And as I said, the pandemic left a big impression on me. And so any opportunity that we have to try to do something to actually impact people's lives is very good to me. And so, yeah.
Gotcha. So when you first started your PhD back in like the early 2010s, was this on your mind already? Like, did you pick this advisor because he was entrepreneurial? Like, what was the thought process back when you first started your PhD?
That's a good question too. And the answer is no. The answer is no. I picked my advisor actually because I thought he had a really wide range of intellectual interest. And when I was a young grad student, I was also had a very wandering mind, if you will. And I find many things interesting. And so I like the idea of working with somebody who is open-minded about topics and approaches to pursue. And yeah, so that's how I started. But I wasn't thinking about starting a company at the end of my PhD at all.
So how did you land on this imaging PhD thesis?
I always had an affinity with imaging. So the first summer research internship I did as an undergrad was actually analyzing star images, X-ray images of stars. And so I was looking for young stars. Yeah, it's very different from what I'm doing now, but that is imaging based because we're taking pictures of stars and trying to analyze their spectra. And so I've always had an affinity for imaging. And in grad school, we had a really required experimental course for all physics students. You basically have to do 3 experiments in a given semester.
Okay.
And one of them was imaging based, and I just, even to this day, I still remember very fondly of that experiment. So I've always had a knack for imaging, if you will, and I was introduced to the microbiome field when I went to a summer course in Woods Hole. So this was at the Marine Biological Laboratory. And so there I really got exposed to the microbial world, and it was just fascinating to see all these tiny little creatures living together in a very structured way. And that was eye-opening and striking to me. And so when I came back, I told my advisor, I want to work on this. And so there we go. We started there.
And because the advisor's open-minded,
Right.
he was like, yeah, this sounds good.
Yeah, and he obviously has been in the field for a while, and he has worked with the microbiome as well, using other approaches to that. And so he also saw that there's a need for new technologies to look at the microbiome in a different way.
Interesting. So like, what was your thesis on? Was it like hardware related on like the hardware of the microscope? Was it more software related on how to analyze the images? Or was the methods? Like, what was the thesis?
It was more like a full stack, except for the hardware, I would say. So it's got molecular biology approaches. It's got computational approaches, methods to generate sequencing data, analyze the sequencing data, designing probes. It's got image analysis methods. So how to actually turn a raw image and quantify it and digitize it so that you can actually analyze it rather than just looking at it. So yeah, it's got lots of different, I wrote lots of software, not good software, it's just good enough for me to do the work I need to. But yeah, it's got lots of different facets to it.
Interesting. So you bought the microscopes. After you bought the microscopes, everything else was all you.
Yeah,
The
we didn't
analysis.
buy the microscope. We just use the microscope at the core facilities. These are
Oh,
commercially.
gotcha. Okay. This is fascinating. What was your thesis?
So my thesis was actually just describing the technology. So it's describing a spatial mapping method that I developed to look at the microbiome.
Gotcha. Okay. Makes sense. So after you started a company, how did the way you approached the problem change? Like, did you have to change your mindset over time?
Yeah, certainly my mindset has changed quite a bit. I think when I was in grad school, I tend to be very focused on technical problems, as probably many grad students are. But at Canvas, I think we have a much broader view of where the technology was going, what we want to use it for, and how does that relate to the clinical programs. And so there's a lot more kind of broader view of what we're building and how it's going to be used. And so I think that's probably the biggest difference, I would say.
Gotcha. Broader as in how it'll save a patient's life in the future?
Yeah, broader in how it's going to fit into everything that we do at Canvas, how it's going to help with building better drugs, how can we actually optimize certain aspects to suit what we need to do on the clinical programs, things of that sort.
So does broader also include like the mouse trials as well? Because before you didn't have to do mouse trials, now you have to.
For sure. We have to do a lot more mouse studies. I didn't really do much of that in grad school because again, I was very technical, focused on the technology aspects, not so much the biology aspects. But here at Canvas, we obviously have lots of biology facing components to actually get a drug to the clinic.
Gotcha. And do you have any tips on how to build up a team to solve the problem that's more broader?
Yeah, I can share what we did, but I don't know if that will be applicable to everybody. So early on, we try to find people that are what we call multivalent, and so essentially very technical people who is good at many things. You can throw them into any situation; they will try to figure out the solution somehow. And so early on, we tried to find teammates like that. As you can probably imagine, as a young startup, there are many different things that needs to be done, and you can't always hire specialists for everything. And so having people who are skilled in many aspects are very helpful. We also like to hire people that are good people to work with, essentially. And that really solidifies a good team dynamic, which then reduces friction along many different aspects of work.
So how has the team grown over the years?
We started as a team of two, I would say, two operators. We are now at a team of almost 30, and we're split about half on the East Coast, half on the West Coast.
Oh, wow. That's great. Did you start hiring like business development people as well? Did you start hiring like more drug safety, regulatory people too? Or like what did you keep in-house and what did you keep out of house over time?
Yeah, so early on, we hired a lot of PhD scientists, mostly on the R&D side, to try to build out the tech for various different applications. We then started hiring BD people. We now have regulatory people. We have drug development experts. So now we really have a very wide range of expertise within the company. But we don't necessarily do everything in-house. So for example, for clinical trials, we'll still work with external CROs because that oftentimes require a large operation that doesn't really make sense for a small startup to completely do in-house.
Yeah, that makes sense. So the mice will be at the CROs then.
Right.
That way you don't have to maintain the mice yourself.
Right. We ask them to run a mouse experiment and then send the samples back. So then we have the samples and we can do any number of experiments.
Okay.
Yeah. They usually send back the tissue. So they would sacrifice the mice and send back the tissue.
Oh, so how do you organize your team? Is it by each each clinical trial, or is it by each product, or like how do you think about the pipeline?
Yeah, we have roughly grouping our team members into two buckets. There's the platform side and there's the clinical side. So on the platform side, we have a few team members that are in charge of various aspects of improving our analytical capabilities, looking at more different types of biomolecules, for example. And then on the clinical side, we have different sub-teams for different clinical programs. And so, for example, we have a team for the CAN001 program, which is for ICI potentiation. And then we have a different team for CAN004 for ICI-induced colitis.
Okay, gotcha. So then colitis is the one that's going to go into phase 1 next year. Right. And then, gotcha. This is great. This is a huge milestone to this phase 1 trial. This is great.
Thank you. Yeah, it's a big moment for us.
Do you think in terms of like baseball analogy, like a single, so you can easily hit this, and then the next one you go for a bigger one? Like, did you go for what was the strategy behind like which ones to go for first?
Yeah, so there are a few different factors that goes into when we launch trials. And so in this particular case, actually, the manufacturing for one of the programs was ahead of the other, and so it
Oh,
made sense to start the one that's basically essentially made to start that one first, and then do the next one after the second batch of drugs have been made.
gotcha. That makes sense. So I keep hearing this new term in biotech, MSAT, manufacturing science and technology. Are you in this area?
I wouldn't qualify us as directly in that bucket, but I think what we are doing in terms of manufacturing is certainly very new in the sense that we have tried to look for external parties to be able to, so it's other CROs to manufacture the drug for us. But what we learned is that no one else has the technologies that we do to really look at a drug in the detail that we can. And so we are actually going to be doing the drug manufacturing ourselves.
Wow.
So in that sense, we are building a lot of technology around that area. But I think it's probably a little different than the classical MAST definition.
Gotcha. That makes sense. The microscope is good at before the pill goes to the patient, you can look at it
Yeah,
because you control the whole process. You can
exactly.
actually look at it
That's right.
beforehand.
That's right.
Interesting. I'm picturing like a pill, like it's like a little white pill with like a little indentation on it, and you just take it. Is it like that kind of pill?
It's gonna be a little more complicated than your typical aspirin pill. So it's actually gonna be a pill inside the pill because we need to make sure that the pill doesn't get chewed away by your stomach acid. And so it basically needs to survive long enough to go to your small intestine before it releases the bugs in the lower intestine.
Oh, so there's a stomach before the small intestine.
Yeah, yeah, yeah.
Gotcha. So you gotta
You gotta
you
make
have
sure your
to.
drug goes through. Yeah, the bugs have to survive the passage through the stomach, and then we release them.
Oh, this is complicated. Oh, wow. It's very innovative in so many ways then.
Thank you. Yeah, it's certainly an adventure. Yeah, everything we're doing is quite new, and yeah, we're very excited to bring these drugs to clinical trial.
Yeah. When you talk to venture capitalists, for example, how do they view it as like a immunotherapy kind of drug, or do they see it as like a whole platform for many
Yeah,
different things
that's
in the future? How
yeah.
do VCs bucket you?
Yeah, interesting question. Or different VCs bucket us differently. And so we have talked to many VCs that are only interested in the platform. We have talked to VCs that are only interested in the program. But luckily enough, many of our key supporters really see the whole picture. They really see the potential of the platform. They also see that the platform has some immediate use for building the clinical programs or helping the clinical programs. And so it really depends on who you talk to.
Yeah, that really de-risks it then if you have the immediate ones. I could see a lot of drugs being manufactured in this way in the future too. So
Yeah.
it could be like a CDMO.
Yes, absolutely. In
Very
fact, we
interesting.
are, yeah, that is one of the things that we are uniquely suited to do. Yeah.
Yeah, especially if you can manufacture your own drug. This is great. Huh. In manufacturing, like you actually have like a little factory somewhere.
So yeah, we are building that up right now. It's gonna be on our San Francisco site, so it will be a GMP facility with all the. I think you can imagine like a brewery, essentially. So lots of bioreactors with valves and such. They will be quite big, and they will be in a very controlled area. So that's kind of what the setup looks like.
Yeah, there'll be like huge vats of microbiome inside, and you have to feed it constantly, and it grows and grows,
Yeah.
and then you turn into little pills, and then people would take it. So for the closing question, Haoshi, I always ask, what's the most kind thing that anyone's ever done for you? What's the kindest thing that anyone's ever done for you?
My answer might not be very satisfying, but I think I am an immigrant, and when I came here, I really don't really know that many people here. I don't have
Mhm.
family here. I think the kindest thing people do is just taking chances on me along the way, and this has happened many, many times throughout my career in undergrad, in grad school, and now in Canvas. And I think I owe a large part of my success to just very kind people taking chances on me, and I hope to be able to pay it forward in some way going in the future.
How old were you when you immigrated?
I was 18, so I came here for college.
Oh wow, super. Thanks for sharing, Haoshi.
Thank you. It's great talking to you.